Hello, I'm Bhalla. I'm a computational neuroscientist at the National Center for Biological Sciences, Tata Institute of Fundamental Research in Bangalore, India. I'm here to tell you about real neurons and the reason why they are such wonderful computational instruments and how they compare with the more artificial and abstract versions that you will have heard about and will hear about in the Neuromatch Academy series. Ramon y Cajal, many years ago, very poetically described neurons as the mysterious butterflies of the soul. Here's a small sample of some of the very beautiful illustrations he made of the neurons that he studied. And the key point from this is that neurons come in a marvelous variety of shapes and sizes, and presumably these shapes and sizes have something to do with their function. We will have a glimpse of some of these functions that arise from shape and other very subtle features inside the neuron, which confer upon the neurons a lot more capabilities than simply integrate and fire, or for that matter, just summation of assorted inputs. So here's a picture of a real brain, and of course this is composed at many levels of neurons and circuits and other components, and this is what we're interested in studying. Now the thing is, this real brain is about 1.3 liters of matter. It consumes about 20 watts of power, and yet it does all of the things, all of the processing, everything that we're so interested in understanding. Contrast it, for example, with this TPU, Tensor Processing Unit from Google, which occupies huge industrial size racks of space. It consumes megawatts of power, and at least so far, it can only do a small fraction of things that the human brain can do, and not very well. So what is it that the real neurons and the real brain are doing that make them so much more effective? Well, just to give you a glimpse of things, here's an example of a typical artificial intelligence neuron, where you have a set of inputs, 1, 2, and 3, each associated with a weight. All of these summed weighted inputs are combined and summed. It goes through input-output nonlinear transfer function to give you the output. So this is the computation, the core computation that an artificial intelligence neuron will do, and you need millions and billions of them in any of these neural networks to do what they can do. What Wilfrid Rall did many years ago was to show that this very reduced picture of a neuron is lacking in some very important fundamental ways which have serious computational implications. His primary contribution was that the actual beautiful intricately branched structure of a neuron cannot be treated in the same way as a spherical point neuron. We will also see some of the crucial work by Hodgkin and Huxley, who showed that the very dynamic, the very complicated properties of firing of neurons cannot be reduced to either simple rate models or to integrate and fire concepts. They looked at Hodgkin-Huxley channels named after them and spiking, and described them in terms of an equivalent circuit. And we will be getting to see a little bit more of how this and their equations, the Hodgkin-Huxley equations, how they confer special properties and waveforms onto neuronal firing. Finally, our third category of heroes for the day is Alan Turing. And among his many other accomplishments, Alan Turing wrote one paper on biology, and it was on how one could do chemical computations. He was particularly interested in how it is that through chemical interactions one can form a huge variety of patterns, both in space and in time. And we will take some of these ideas on in this brief introduction to give you a sense of how you can do computation with this in a neuronal context. The brain, of course, operates at all of these levels simultaneously and in an extremely tightly integrated manner. This is a very old classic slide, and the key point of it is just as true today as it was when the slide came out some 20-30 years ago, The point being that computation happens at all of these levels, from molecules, synapses, dendrites, neurons, circuits, areas, systems to behavior. That it is the interplay of signals between each of these levels that contributes to neuronal computation, and that you focus on one to the exclusion of the others at your peril. You will miss out some of the crucial factors of computation in the brain. And so this is something that we will explore today. In a somewhat rough match to these levels of description, here is a set of equations which I like to inflict upon my students, describing roughly at the level of molecules and synapses. Here are some chemical term forms that describe how molecules react and their kinetics. Here is, of course, the diffusion equation, the Nernst equation. Here's the cable equation, which belongs somewhere down there. Here are the Hodgkin-Huxley equations, and up here is a standard form for describing synaptic transmission. And it's quite remarkable how much of the properties, how much of the physiology in the brain at all of these levels can be accounted for by this rather small set of equations. However, this is biology, and it is in the parameters, it is in the connections, it is in the detail of all of these equations that the amazing properties of neurons come. And so we'll be seeing a little bit of that now. So let's start with our first hero, Wilfrid Rall. So, as I said, what Wilfrid Rall did was he explained why it is that real neurons are very different from this kind of artificial intelligence. Well, he didn't have the concept of artificial intelligence neurons in his time, but this kind of neuron is not a good description of what he is looking at, what he tried to describe to us. In fact, the integrate and fire model neuron has a lot more to do with what Rall was looking at, though he actually had more sophisticated views of this thresholding and firing part. But let's start with the core of this, which is the equivalent circuit, where you have a membrane. This is the inside of the cell, this is the outside of the cell, that's the ground, which indicates it's the rest of the cytosol. The membrane has, because of it's an insulator between the inside and outside conductors, it acts like a capacitance. There's a membrane resistance, there's a membrane potential, and you can have various synaptic inputs which are represented as variable resistors in series with some kind of a potential. And in the integrate and fire neuron, what happens is that when you give inputs to this, when this resistor passes current, it might cross a threshold. If it does, then the output action potential goes out, and it internally shorts out this circuit, it resets it, clearing out the capacitor and allowing you to start again for another cycle. And you'll be hearing a lot more about what you can do with such neurons. So what Rall did then, as I said, was to explain that real neurons are much more interesting than these abstract concepts, that the space matters a huge amount. And let's look at that. So here is one of the core intuitions, one of the starting points for thinking about how signals propagate in cells, in extended cells. And this propagation happens both along axons and along dendrites. In fact, any cable that can be described as a conductor with an insulator around it, and then a conductor around that. So here we go. If we give a sharp stimulus at one end of the cable and record what happens to the potential, then at the site of injection of this current, one will see a fairly sharp signal. And then as you go along and record at different positions along the dendrite, you will see responses that change. And what you see are three main things. First, most obviously, the amplitude of the signal is decreasing. It starts out high and it gets lower and lower as you get further away from the stimulus point. Not surprising. If you look closely, you'll see that the shape, which starts out nice and sharp, becomes smoother and wider and broader. And finally, if you look really closely, you will see that the peak of each of these waveforms is getting slightly delayed, slightly offset in time as the signal marches along down the dendrite. Okay, so what can you do with this? Well, what you can do is to hope that the thing will advance, and it doesn't advance. Okay, this thing has gotten stuck. Okay, there we go again. All right, so one can summarize a lot of the theory that Rall came up with in these two very, very simple plots. And basically what it says is that the signal decay, both along time and along space, is a simple exponential. So if you hold the potential at a certain value at time 0 and ignore space, in other words, treat the cell as a point object, and then ask how the potential evolves with time, it turns out that it evolves as a decaying exponential with a characteristic time course tau. If, on the other hand, you ignore time, that is, you take time at infinity, at steady state, and you ask the same thing of the potential along distance in this case, you hold one end at a certain potential, and then the potential along the cable's length will decay in an exponential manner with a characteristic length of lambda. So both in time and space you have an exponential decay, which is a nice straightforward way to think about it, and these are both solutions that come from the cable equation. So, of course, among many, this actually predates Rall. People had worked out the cable equation even before he did, but he applied it in some very interesting ways. Here's one of them. So what Wilfrid Rall did was to come up with a description of how a single neuron could do a rather interesting computation, that of sequence selectivity. And it works like this. Suppose you have a nice long neuron, a nice long dendrite culminating in the soma, and you give a sequence of inputs in order 1, 2, 3, 4. So let's consider what happens with the first input. You give an input here, and just as you saw earlier, it leads to an initial sharp peak. There's a spreading and a decline in amplitude, but importantly for this, there's also an increase in time to peak. The time it takes is a little bit slower than at the initial one, and this time keeps increasing. Now, suppose your second input comes just when the input from the first location has reached its peak. And then you apply your input here, and now these two peaks will coincide, and they will coincide as you go along down. And you can put your third input now just when these two peaks have arrived at this point, and you get yet more summation, and so on. And so if you time your successive inputs in such a way that the peaks all coincide, then you can get a nice large response. And this kind of a situation might come up, for example, if this neuron were the recipient of input from a number of photoreceptors, and let's suppose that there is a fly, an object, zipping past the field of view such that it activates this photoreceptor, then that, then that, then that, and then these all contribute to the signal here, and you get a nice large signal. Note that with this arrangement, if the fly or object were going in the other direction, you would not get summation, you would not get such a large peak. And this is, in fact, something which was recently experimentally validated by Tiago Branco in Mike Hauser's lab, and they did it like this. So here's their neuron, nice, beautiful neuron with lots of dendritic spines. There's a recording electrode, and there is the piece of dendrite that we are interested in. So what they did, so here's a zoomed up picture of it in B. What they did was they used a technique called glutamate uncaging to release neurotransmitter in this particular order, 1, 2, 3, 4, 5, 6, 7, at these specific locations, and ask what happens to the signal as recorded at the soma. And what they found was if the sequence of inputs delivered is in the same order as numbered here, in other words, if the stimulus comes towards the soma, if the sequence of inputs comes towards the soma, then you get a nice large response in red. However, if it goes in the opposite direction, then you get a shallower, broader peak. And this is exactly what Wilfrid Rall had said, that if you get the sequences of inputs coming towards the soma, you get a big peak. If it goes the other way, you get a shallower peak. And what they further showed was that the sequence selectivity is also velocity selective. That is, if the speed at which successive inputs impinge upon the dendrite is at a certain rate, say 4 microns per millisecond, then you get the largest possible difference between the inward going and outward going sequence of inputs. And so you get nice strong sequence selectivity over here. And so this is a confirmation of Wilfrid Rall's prediction from many, many years ago. This has some more subtleties, for example, the role of the NMDA receptor, which I won't get into today. So the point here is that here you have a single neuron with just the standard physical properties, biophysical properties underlying it, and it's performing a rather sophisticated spatiotemporal computation to determine sequence direction. So now let's move on to our next heroes, Hodgkin and Huxley, and here they are. And what they did, of course, was to describe an action potential. And what we're going to do is we're going to spend a little time designing an action potential according to the principles that they laid out. So all of you are familiar with this, but I'll recap it anyway. When you deliver a charge to a cell, it causes a slow depolarization. Here we're using original Hodgkin-Huxley units starting at 0 millivolts. So you depolarize the cell, eventually you hit a threshold, the sodium channel opens, and you get a nice large peak. At some point, the potassium channel kicks in and the sodium channel starts to close, and the membrane potential starts to plummet. As you go a little bit further along, the sodium channel is shut, and now it's a potassium channel, and it is pulling the potential even more negative than resting potential. And then if you wait long enough, all the channels are shut, and the cell is back at resting potential, and it can repeat the cycle. This can be described again with the equivalent circuit I introduced a little while ago, where you have your membrane capacitance, you have your resting membrane potential, you have the standard membrane resistance, and then you have the conductances due to the potassium channel and the sodium channel, and these are variable conductances, and they depend on the membrane potential, which is why you get this feedback effect. And there's much more detail on this, and I won't go into that. And here, just for the sake of remembering Wilfrid Rall's contribution, you also have other compartments to the left and right of this compartment, and so your current can also flow left and right. Okay, let's look a little bit more depth at what you can do with Hodgkin-Huxley channels. Yeah? So the shape of an action potential is actually crucially important in the function of neurons, and not just anywhere. For example, if you mess up the shape of the action potential in neurons and muscles in your heart, there's the unfortunate side effect that it might kill you. So the shapes of these action potentials can be very, very important. They are also very important in determining what exactly happens with the input-output transfer function and various other things that we'll look at in just a moment. So what happens during a nice complicated action potential which includes these channels? Well, one thing is that when the action potential is near its peak, you have the opening of a whole bunch of channels, typically calcium channels, which introduce calcium, and we'll see what that does in just a moment. Here is the regular hyperpolarizing effect of the delayed rectifier potassium channel, the one that Hodgkin and Huxley described. And I won't go into this just yet, that's the next slide. But let's think of what happens when the action potential is thinking about going in for another cycle. So normally the cell would start to depolarize and the potential would rise rapidly. But in comes our friend Ka, the after hyperpolarizing potassium channel. And what happens is that this channel opens just about the time when the cell is recovering from hyperpolarization, hence its name. So its region of activation is indicated in green. And what that does is, because the channel has opened, this is a potassium channel, it pulls the potential of the cell more negative and it delays the next action potential. Yeah? And so this slows the arrival of the next action potential, and in fact the presence of this channel tends to linearize the firing of a cell as a function of input current. Here's another similar effect, but mediated in a different way by a different channel, by the inward rectifier. What the inward rectifier does, as its name might indicate, is that it opens when the cell is below its resting potential. So it's an inward rectifier. It only opens at one phase of the potential, and it is going to pass current so as to, again, hyperpolarize the cell. And it is active in the region indicated in red. So the cell has become more negative, the inward rectifier channel opens, whereas earlier in the thick line, the cell might have recovered its potential rather quickly. The inward rectifier sort of keeps the cell hyperpolarized for a bit longer, and then again the effect is to delay the presence of the next action potential. So these are some of the things that happen with the range of ion channels that cells manifest. Here's another example. Here we're looking at what happens with the interplay of calcium ion buildup with action potentials occurring in bursts, and something called the calcium-dependent potassium channel, of which there are many. And there are also other channels which may also behave in a similar way. So what do you get? Let's suppose you have a series of action potentials, and as each one comes in, there's a burst of calcium that comes in because the calcium channel opens at the peak of the action potential. Calcium builds up. As calcium builds up, we slowly open the calcium-dependent potassium channel, and this just clamps the cell down. It prevents it from firing anymore, and so what was a vigorous burst suddenly peters out, and then you have to wait for a while for the calcium to decay down to resting levels, and then the cycle can start again. And bursts, as you will know, are very important in neuronal activity and have a particular role to play in plasticity as well as other kinds of signal flow. So here are just some examples of the complexity that comes up and has a computational role in neuronal function when you consider the biophysical details. There are other aspects to this which I won't go into much detail. For example, Eric de Schutter has shown that it's not just a case of which channels they are, it's also very important to know where they are, and their presence and localization along the complicated dendrites of a beautiful cell like this Purkinje neuron has a huge effect on the fact that the cell can have different potentials happening at different parts of the neuron all at the same time. And these mean that the cell can do different kinds of integration in different regions, and all of that then feeds to what the soma decides to do. And it is the precise placement of these channels that is very important. One of the other things that happens because of the distribution of these channels is that, for example, inputs coming as far away as this tiny dendrite way over here may have the same effect due to amplification by these channels at the soma. Oops. It may have the same effect due to amplification at the soma as an input coming over here. And that's because, again, of the role of these active Hodgkin-Huxley type ion channels. Of course, here's a much more dated, if you like, concept that the voltage-gated ion channels do. And this is, in fact, what Hodgkin and Huxley themselves worked on. They worked on the squid giant axon. And this is the role of axons and these ion channels in allowing long-distance propagation of signals. So the squid giant axon is really a mammoth piece. This is 0.5 millimeter across. These little circles over here are the other axons, so you can see just how much metabolic effort the squid invests in this giant axon. Its diameter of this axon is much bigger than the entire cell size of many neurons. So what this does is it allows the squid to communicate a little bit faster because the rate of action potential propagation gets larger as you have a larger diameter. But of course, along at some point came the vertebrates, and they came up with a much better idea, which is to myelinate, which is basically to insulate the axon. And instead of having a massive great fiber conducting information along, they have insulation and very strong amplification at what are called the nodes of Ranvier. Here's a picture of that. Here's the axon, and that's layers and layers in black, layers and layers of this insulation called myelin. And now the signal sort of jumps from one node to the next, and therefore propagates much, much faster for much, much less metabolic cost. Okay, so these are all details, and they arise from the details of the biophysics of a neuron. Finally, let's go to Alan Turing, who talked about chemical computation. So the key idea here is that just like we've been talking about electrical signals as conveying information, we can use chemical signals to convey information as well. And the nice thing is that there are many, many, many chemicals. Yeah. So each chemical, each identified signaling molecule could carry a different signal. And we will later consider spatial separation, which in fact hugely scales up the amount of information that you can process through chemistry, because the length scales of chemical spread of diffusion are much, much shorter than the length scales of electrical signal flow, which means that you can compartmentalize chemical signaling very, very precisely. So here's a classic example of chemical signaling, logic at the level of molecules. And this is something that you are probably all familiar with, which is the associativity due to the NMDA receptor. Associativity means that it acts like an AND gate. That is, you have to have presynaptic activity and postsynaptic activity, and I'll explain how. So Supposing here comes a presynaptic signal, it causes release of neurotransmitter. What happens then is that you have your regular AMPA receptor opening up, and that can pass some signals in, but the NMDA receptor is going to stay closed. And the reason is that the cell is still sitting at a relatively negative potential, and there's a magnesium ion which comes along and says, I'm positively charged, inside of the cell is negatively charged, therefore I want to go in. And it tries to go in, and it goes and plugs the pore of the NMDA receptor. Okay, so that prevents any signal from going in through the NMDA receptor. However, once you've had enough action potentials coming along, or for example, the cell itself fires an action potential and that propagates back up the dendrite due to the voltage-gated channels we were telling you about, you can have the cell now sufficiently depolarized that the magnesium ion is expelled from the ion channel, from the NMDA receptor. Now, when the neurotransmitter comes along, the channel will open and calcium can come in, and all sorts of good things can happen after that, including potentiation and so on. So here you have an AND gate. You have to have synaptic input, you have to have the neurotransmitter, and you have to have depolarization postsynaptically. And this is implemented through molecules. There's an immensely complicated network of molecular signals operating at the synapse. Here's something that I've worked on for years and years, just a small sample of the various signals and pathways that are present at the synapse. This is what it used to look like on my screen decades ago. And of course, now we know far more molecules, and it is even messier on my screen. But that's not the whole story. Here's an absolutely gorgeous visualization of the packing inside a single presynaptic bouton. This is about half a micron from there to there, and you can see the little ornamentation of surface proteins. It's absolutely jam-packed with proteins of different shapes and sizes, vesicles and so on. And the thing to remember is that potentially almost all of these molecules can serve a computational function. Let me illustrate that with the example of memory. So, going way back into an abstract view of things, consider that you have a system of molecules where molecule A activates molecule B, and molecule B activates molecule A, a feedback loop. Yeah, this is a classic motif for signaling and computation. So in the resting state, A is off, B is off, and you don't have any activity. Now let's give it a strong stimulus. A goes on, B goes on, and so A turns on B, B turns on A, and this state of activity continues indefinitely. And you can now break it up. Instead of the bulk, you can break it up into the individual molecules which turn each other on. Okay, now let's transplant these inside a schematic of a dendrite. So here's the schematic of a dendrite. Here are dendritic spines, these mushroom-shaped blobs, and we start over here. Yeah. So here, let's say, for the sake of argument, that the triangles and circles are in a positive feedback cycle and they're on. Okay, well and good. They're storing information perhaps about a previous event. But as time goes on, the molecules start to die. They degrade. And what happens is that if this were to continue, you would keep losing molecules that hold the signal, and you would lose the memory. And this is not a good thing. So, fortunately, the cell is alive. And because it's alive, it makes new molecules. But the key thing is the new molecules do not know anything about the state of the existing molecules. They come in, they're naive. But because there's a lot of activity here, this active one will turn that on, those active ones will turn that on, and pretty soon the new molecules have been recruited, so to speak, to be part of the memory. And in this manner, you can turn over all of the molecules that are holding the memory and yet retain the memory itself. Yeah? And this is really important because the lifetime of individual molecules of this kind in the synapse is measured from minutes to maybe a few days. And one really hopes that one's memories last longer than that. So let's now wrap up with a view of scales. I really want you to put this in perspective so as to get a sense of the sheer computational capacity that we have going from molecules to biophysics to the whole neuron. So this is about half a micron across. Yeah? This thing packed and packed with protein molecules which are doing computation. Yeah? Okay. Now we go way larger, 100 times larger, and here we have a 5 micron cube. Yeah? This is from a reconstruction by Kristen Harris and colleagues. And the synapse that we looked at earlier is that big. In fact, here is a dendrite down the middle in yellow, and these little red dots are synapses being made onto this dendrite, right? So the synapses are really tiny compared even to a small segment of dendrite. Let's now zoom out again a bit more. So that entire chunk of dendrite that we were seeing with hundreds and hundreds of synapses is actually just a teeny piece of this beautiful complex Purkinje neuron. It's just a tiny part of what's happening on the scale of the cell as a whole. And now we'll zoom out again, and now this whole neuron fits in this tiny little piece here back in the cerebellum, because that's where Purkinje neurons live. And so I hope I've given you a sense of orders and orders of magnitude of potential computation that allow the cell to do what it does so efficiently, and therefore allows the brain to do what it does so efficiently. So just to give you some sense of numbers to wrap up, the brain has roughly 10 to the 11 neurons in it. Each neuron has something like 10 to the 3 or 10 to the 4 synapses, for a total of 10 to the 14 or 15 synapses. Each synapse has, I don't know, maybe 10,000 reactions going on. So a lot of computation happening at the molecular level, and of course a lot of electrical stuff happening. Electrical stuff is fast, of the order of a millisecond. Chemical events are slower, but I remind you that synaptic transmission, which is primarily chemical, also happens at the millisecond time scale. So chemical events take longer, structural events take longer still. The lifetime of a protein, which I told you about, degrades, that is of the order of days, but the lifetime of a neuron is all your life, and hopefully so is the lifetime of a memory. And all of these things are happening because all across these scales information is flowing back and forth, and a huge amount of computation is happening right down at this most microscopic, submicroscopic level. Okay, to summarize then, networks, which are what we're now in the era of deep learning and so on, we treat as a dominant metaphor for brain computing, but they're very, very inefficient. And the reason why the brain can accomplish what it does in such a small scale of size and such low power consumption is that it's doing things very efficiently: summation, integration, computation of sequences, and many other things, association and memory. These are just a few of the examples which I've given you about what the neuron is doing with absolutely minuscule costs in computation or in size. And as a take-home message, if you wish, when you do your modeling, when you try to understand what the brain is doing, it's not necessary that you dive into the most ghastly detail of what individual molecules are doing. You have to model at the level needed for your questions. For example, if your questions relate to disease or development or synaptic plasticity, turnover, injury, then there's no choice. You have to include the molecules because this is where the action is for such phenomena. If you're interested in cellular integration, some kinds of sequences, fast dynamics, then you should probably include the biophysics. And just to remind you, computation occurs across all of the scales from the molecular to the electrical to the whole cell and firing and network levels. These are all part of it. They're not something that you can home in on just one bit and ignore the rest. And I think that cross-scale effect is precisely what makes the brain so efficient and what makes it very interesting to study the properties of real neurons, and you'll be getting to hear more of this in the coming lectures. Thank you.